Our next speaker is Dr. Greg Holland, professor at San Diego State University. Dr. Holland received his PhD in physical and analytical chemistry from the University of Wyoming. He then became a postdoc at Sandia National Laboratories in the lab of Todd Allam, where he focused on NMR spectroscopy of biomolecules and nanomaterials. Following his postdoc, Dr. Holland was a research professor in the Magnetic Resonance Research Center and Department of Chemistry and Biochemistry at Arizona State University. In 2015, he moved his research group to San Diego State, where his lab continues to focus on the development and application of magnetic resonance for the characterization of biologically inspired materials, molecules, and nanomaterials. His talk today is Feeding Spiders Isotope-Enriched Amino Acids to Labor Their Silks for NMR Investigation. And Greg, the floor is yours.
Sorry, guys, I forgot to unmute.
No worries. We cannot see your screen either. Yep, there we go.
All right, you hear me okay?
Yep,
See my mouse?
looks good from our end.
All right, great. Thanks, Kelly, and thanks for the organizers for having us. It's been a really great webinar so far. As you can see on my slide, it really just puts in perspective what I'm going to talk about today, which is, my lab. One of the biggest projects is the structural biology of spider silk, I would say, and how spiders produce silk. And one of the ways we get after this is by NMR spectroscopy. And so the goal is to label the silks and the fluid that the spiders spin into silk inside their abdomen with isotopes. And this is the spider's mouth here. And we literally collect silk from the spiders and dropwise feed them in their solution of different carbon-13 and 15. We've done deuterium as well, labeled amino acids. And we look at both ends of the spectrum. So when I came into this field, my training is in solid state NMR. So we looked a lot at spider silk fibers, but more recently, we've been actually looking at the internal workings and the silk producing process as a whole. So we'll label the spiders with isotopes and then dissect the spiders and actually remove the fluid and use solution NMR to look at that. So we can look at the silk, pre fiber assembly. So I'm just going to point to a few people up front in the acknowledgement. So two senior people in my lab, Dr. David Onofre, who's a research scientist. He runs the NMR facility at SDSU, and he's an expert in solution NMR. So he looks at the fluid before it's spun into a silk fiber. And then my postdoc, Dr. Kevin Chalik, is an expert in solid state NMR. So he looks at the endpoint where the silk has been spun into fibers in fiber form. And then Hannah Johnson, who's been looking at liquid-liquid phase separation. Liquid-liquid phase separation is a hot topic in biology, and we think it's an intermediate step in spider silk formation. So she's been looking at processes of liquid-liquid phase separation. And a number of collaborators. I'll mention a little bit at the end some work we've been doing with Galia Develachina at UCSD using DMP, magic angle spinning solid state NMR. We also do a lot of simulations, which you'll see dotted throughout. And the funding for the spider silk work comes mostly from the Department of Defense, Army, and most of what I'll tell you about today has been funded by the Air Force. And so one of the things that a lot of people don't realize when talking about spider silk is that spiders don't produce one type of silk. They produce a range of different silks. And so they can produce up to 7 different types of silk. And so the interest in silk has to do with its mechanical properties. So the dragline silk or the major ampulate silk, which is the framework of the web, also the safety line. So if you ever see a spider dropping from the ceiling, that's the dragline silk. Its mechanical properties are shown here on this stress strain curve, and this is 3 different species of spider. And you can see they vary, but they have similar responses. It's stronger than steel by weight. It's tougher than Kevlar. But prey wrap spider silk, which is the one that's shown here, which if you've ever seen an insect in the corner of a web to be eaten later, and it's wrapped in a silk, that's prey wrap silk. And you could see its mechanical properties are very different. So much more extensible and actually tougher than dragline silk. It also has an interesting interaction with water where it cross-links and changes its conformational structure. And I mentioned the prey wrap, there's also egg case silk. So they use prey wrap silk in egg cases, and they also use a primary component called tubuliform silk. And tubuliform silk is actually stiff, almost like a glass rod. You could actually flex it. So they make a whole library of materials. There's one called flagelliform silk, which is the spiral of the web, and that has elasticity like rope. So they make a lot of different materials, and they vary tremendously in their mechanical properties. What I'm going to tell you about today focuses on the dragline silk. The other thing I mentioned, a lot of the funding comes from DoD. The Department of Defense has a long history with spider silk. So actually, in World War II, the Army actually had sheds of black widows that they collected silk fibers from for the gun sights because they needed, a 1 micron perfect cylinder, and this is all pre, polymer fiber chemistry, and so they actually used spiders to create those gun sights. And then, of course, if you could ever do it at mass, at scale, you could imagine developing all sorts of different types of materials for everything from the DoD to the biomedical, drug delivery. Silk has been used as a suturing material and in wound healing applications for a long time. So there's a number of different applications for silk. This is 2 of the proteins that make up spider dragline silk. So the major ampullate silk, you could see that they're huge and they're highly repetitive, and that makes them very challenging to study in general, but of course by NMR as well, because you don't have a lot of the chemical shift dispersion that you would if you have all the 20 amino acids represented. So there's about only about 4 amino acids represent about 75 to 80% of dragline spider silk. The other interesting thing about spider silk is that, regardless of which silk fiber you're talking about, and in this case, it's the dragline silk, there's 2 primary proteins that make up the silk that are shown here, MASP1 and MASP2, but there's actually a number of other different proteins in there as well. They're in much lower abundance, but there's more than just 2 proteins. So it's a very complicated system. And when they've looked hard enough, in some cases, they've found up to 15 or 20 different proteins. So a lot more is known about the fiber structure. So after it's spun into a fiber, and this is information from solid state NMR, and what's well known about silk, and not just spider silk, spider dragline silk, but also silkworm silk as well, is that it has a dominant beta sheet component. And we've been able to figure out the identity of the beta sheet components, these polyalanine runs that form the beta sheet component, and sometimes these flanking regions of glycine and serine are also incorporated in the beta sheet. A lot less is known about the GGX region, where the X amino acid is typically something polar, like glutamine, serine, or tyrosine, but it forms approximate 3-1 helical structure, but it's much more disordered. It's non-crystalline. And then the second spider silk protein actually has proline in it, in a GPGXX motif, which forms beta turn structures. Looks a lot like a type 2 elastin-like beta turn. And so what I'm going to spend most of my time today, so that's about the final fiber structure, but what I'm going to spend most of my time today is talking about the fluid that the spider synthesizes inside of its abdomen. So if you look inside a spider's abdomen, this is the major ampulate gland. There's the tail region where they synthesize the proteins, and then there's the gland region where they store the proteins, and then there's a duct region, which is way longer than it needs to be to get to the final endpoint, which is the organ called the spinneret, where they extrude the silk. And there's lots of biochemical changes which occur in here. There's a pH gradient where the pH gets more acidic. There's an exchange of different ions. There's introduction of potassium and phosphate and removal of sodium and chlorine. And so, of course, when you think about changing different biochemical conditions, you think about solution NMR as being an ideal tool. And so we can label our spiders and get the fluid out and do solution NMR studies on them. And then, of course, at the end point with solid state NMR, we can look at the final fibers. So the research in my lab, natural materials, and whether it's spider silk, we also have some projects on bone. Natural materials are really good at hierarchical assembly, right? So they can control things from the atomic all the way up to the macroscopic. And so we use a lot of different techniques to touch in throughout to get on to the different length scales. But most of what I'll tell you about here is using solution NMR to understand the structure of the proteins before they're spun into fibers. I'll mention a little bit about liquid-liquid phase separation, which we think is an intermediate step in spider silk formation. And at the end, I'll touch on some progress in solid state NMR, understanding the structure of fibers. So the structure of the protein before spinning in the gland has been something that's been up for debate. They look a lot like intrinsically disordered proteins, and I'll show you some NMR data here in a minute, and that'll become apparent. But some small angle X-ray that's been done by some of my colleagues and others have indicated that the protein in the gland looks partially folded. So it doesn't look completely folded, and this is showing some small angle X-ray data, but it looks partially folded, and you can see that over here in the cracky plot from the major ampulotope. It looks like this partial sort of folding, but by NMR, and I'll show you some data, it looks mostly like an intrinsically disordered protein that's highly dynamic, and we've done a lot of modeling. I point to the full-length proteins. You can't do atomistic modeling. These are 300 kilodaltons in size, and this is some Martini modeling, but we see some sort of anisotropic shape, a tubular-like structure, and if you do some finer modeling to look at like atomistic models with Charm 36, you could see what you get is a lot of beta turns and bends, mostly unstructured, but lots of beta turns and bends. There's not a lot of helix, and there's not a lot of beta sheet. And so this idea that there's lots of beta turns and bends could explain how it could still be very dynamic and look like an IDP, but still compact itself to give the SAX results that you see above. So what we do is we feed the spiders isotopes, and then we dissect them and we remove the gland. Sometimes we take the fluid and dissolve it in some solvent. Sometimes we take the glands actually directly. You can see them here in a Shigemi NMR tube. And we feed them different isotopes to target the different regions of the protein. I give a little reference here. We've done some work to actually track where the different isotopes go. The basic sort of rules apply of when you're feeding isotopes to cells, right? Like if things are involved in different biochemical pathways, certain ones get jumbled. If you have things in rings, they like to stay together. But if you want to do something like feed selective do selective isotope labeling of something like alanine, and you don't want it to get converted to glycine or some other amino acid, you could just feed unlabeled amino acids along with the labeled one, and this basically works. And so I'm not going to go through a lot of the details. Over the years, we've gotten very good at getting good isotope enrichment. So this is what a basic HSQC N15 HSQC looks like of Black Widow dope, and this is ones that we've just fed with alanine, and you can see, this is the span of the amids, and it looks a lot like an intrinsically disordered protein. I showed GP1, which is a well-structured protein over here, and you can see the narrow chemical shift dispersion, right? It's less than a half ppm, and we've done all the relaxation stuff, and I'm not going to get into that. They're very dynamic, and they look like IDPs. But one of the big questions is, well, how do you rationalize that it looks like a completely unfolded protein by NMR? But small angle X-ray seems to indicate that it's at least partially folded, and whether or not you have these sort of beta turns present. And one of the main things to do in solution NMR when you're studying IDPs, when you don't have a lot of chemical shift dispersion in the proton dimension, is go to X-detect methods. And so this is a sample that was a spider that was fed C13, N15 alanine, and phenylalanine. And we do these X-detect experiments at UCSD. They have a nice X-detect probe at 800 megahertz. And so you throw your proton dimension out. You do multidimensional experiments with carbon and nitrogen dimensions. And you can see that you can really improve the resolution. And so I'm not going to bore you with all the different experiments that we've done. What I'm going to say is that we've been able to assign a 15 residue repeating unit, and we've been able to get all the chemical shift information for all the relevant backbone and C beta side chain chemical shifts. And this is the repeating unit that we've been able to assign. It's GQ, GGA, GA, and then this polyalanine run. And so it's highly represented throughout the sequence. So it occurs a number of different times, and it represents about 20% of the sequence. So of course, it's not, the complete sequence, but it's getting there, right? And so we've been able to then take all this chemical shift information and put that into chemical shift Rosetta. And so we heard about the RNA Nobel Prize. David Baker yesterday just won the Chemistry Nobel Prize for some of his work in computational chemistry, along with the AlphaFold guys. And chemical shift Rosetta is something that he worked on together with Ad Bax that combines chemical shifts together with the Rosetta energy minimization, modeling software package. And what we've been able to do is be able to produce some structures, and you can get some converged structures, and those are 15 residues. And we've been able to expand this to 120 residue structures. And what you see is almost no beta sheet, almost no alpha helix, but you see unstructured regions are the most common, which is not surprising based on sort of the NMR response, but you see a lot of beta turns and bends. And a lot of our simulations were indicating there's lots of beta turns and bends. So even though it's highly dynamic and it looks like a classic IDP, you can imagine getting a very compact structure which still exhibits a very rapid dynamics. And so we've been putting this together with some of the small angle X-ray work that we're doing ourselves, together with the NMR, to get a better understanding of the overall structure beforehand. And so I just want to touch on a couple of quick things at the end. So I mentioned liquid-liquid phase separation as an intermediate step in spider silk formation. So there's an introduction of phosphate and potassium, and you can actually do this, ex situ in the lab, pull the fluid out of the spider and expose it to the different biochemical conditions that we think is responsible en route when it's moving through that ductwork to form the final fiber. And these were samples that we prepared where we fed C13, N15, arginine, and tyrosine, because we were very interested in the role of these cation pi interactions. And I don't have time to go through all the data, but I just mentioned that we see some significant perturbations in arginine, and we're starting to understand liquid-liquid phase separation as an intermediate step en route to spider formation. These same sort of samples, so thinking about it in terms of a solution NMR, we've done solid state NMR where we've done dynamic nuclear polarization, magic angle spinning. So this is to enhance sensitivity. So this is still pulled from the same, done the same feeding protocol that I just showed you for the liquid-liquid phase separation. And this is more work on building our model in spider silk fiber structure. And we see cation pi interaction between arginine and tyrosine. And so we're using that as a way to sort of connect what's happening during liquid-liquid phase separation to what you actually see in terms of the molecular contacts in the final spun fiber. And then the last thing I'll mention, again, in the same sort of vein of understanding sort of side chain interactions in spun fibers. So, the backbone secondary structure of spider silk that I mentioned, the beta sheet at the beginning, and these turns and these 3-1 helical domains are sort of well known, but what hasn't been known is sort of how the side chains organize together. And that's what we've been really focused on. So the arginine tyrosine interactions, and then this is labeling where we targeted MASP2, which has proline. So MASP1 doesn't have proline, only MASP2 has proline. So you can feed proline and also tyrosine as well. And we were looking at ring interactions between the proline side chain and the tyrosine aromatic ring. And this is a way of measuring distances in solid state NMR. It's a DAR method where you can selectively excite the tyrosine ring and then watch build-up curves to the proline ring and do things like estimate distances between them. And we combine this with some of our solid state NMR stuff together with atomistic molecular dynamics simulations with collaborators to understand how these interactions occur. And what you can see here, what's straightforward from these build-up curves is that you could see a much closer distance for the C gamma of the proline to the aromatic ring. And so proline can actually pucker itself. And so you can actually see from these different buildup curves that there's actually a pointing of the C gamma towards the aromatic ring. So that's just another example of how we're building on our understanding of spider silk structure in the fiber form where we're focused on side chain interactions. And with that, I'll be happy to take any questions.
Wonderful. Thank you so much, Greg, for the excellent talk. There is one question in the Q&A box from an attendee. Would it be possible to feed the spiders D2O along with 13C amino acids in order to presumably get higher resolution solution state NMR spectra?
Yeah, so feeding D2O does not work. They don't like that. But we have done, and we haven't applied it to solution NMR, but we have done it in solid state NMR, feed deuterated amino acids. So that's a great point, that you could feed, deuterated amino acids to improve the resolution. Yeah, so that's a great idea. So we've done a lot of deuterium NMR to understand sort of dynamics in fibers, but we haven't done it in solution NMR. But that's an excellent idea.
Wonderful. Well, thank you so much once again for a wonderful talk. We appreciate it. Very cool work you guys are doing.
Yeah, thanks.